In this second part of Chapter 3, we will be looking at the plasma membrane. So the plasma membrane is this barrier that surrounds and contains the cytoplasm of the cell, and it can be very flexible but also sturdy at the same time. It's made up of about 75% of a phospholipid bilayer, and what this means is that it is made up of a dual layer of phospholipids. You have an exterior layer and an interior layer. And this occurs because these phospholipids are amphipathic. Remember that amphipathic means that they have both polar and nonpolar parts. Remember that the heads, which are on the outsides, these are going to be very polar or hydrophilic or water-loving, whereas the tails, which are going to be on this interior part, these are going to be very nonpolar or very hydrophobic or very water-hating. So because you have water or very polar compounds on both sides of the plasma membrane, you're going to have the heads that are going to be facing the interstitial fluid on the outside here, and also the intracellular fluid on the inside. So you're going to have those hydrophilic portions, hydrophilic heads that are going to face the water, and then the hydrophobic regions or the tails are going to be on the inside so that they never touch the water portions. 20% of the membrane lipids is cholesterol. You can see these little orange bits in here. These are going to be the cholesterol. And it wedges its very plate-like hydrocarbon rings between these phospholipid tails. And these cholesterol works to stabilize the plasma membrane. So this decreases the mobility of the phospholipids and it decreases fluidity of the membrane. So when we have an increase in cholesterol, that equals a decrease in mobility. And this is just mobility of the membrane itself, so it's not as fluid. It's more of a rigid kind of membrane. About 5% of the plasma membrane is glycolipids, and these are lipids that are attached with sugar groups. So here are going to be these glycolipids, and these are only found on the outer plasma membrane surface, so they're not found on the interior at all. There are 5 functions of the plasma membrane that I'll quickly go over. So the first one is that it is a mechanical barrier. So this is just a physical barrier that keeps things out. The second one is going to be a selective permeability. Selective permeability. And what that means is that it doesn't allow everything to go through the plasma membrane. It's very selective in what it allows to go through into the cell's interior. And we will kind of go a bit more into detail with that later as well. The third one is that it produces this electrochemical gradient. And this is a gradient, which means that there is a difference in the charge on both sides of the membrane. So you could have like a more positive charge on the outside of the membrane than on the inside. So that's the chemical part, or sorry, the electro part rather, of the electrochemical gradient. And the chemical part is that you're going to have more molecules of one kind on one side than the other. So say you have lots of sodium on the outside of the cell and not as much sodium on the inside. So it produces this gradient or this difference in concentrations on either side of the membrane, as well as a difference in charges on either side of the membrane. Again, we will go into more detail of the electrochemical gradient a bit later in the lecture. Fourth one is communication. So this is a big role that the plasma membrane has, communication with other cells. And that does so with these proteins that stick out on the outside of the plasma membrane. It's used a lot in communication with other cells. And the fifth one is cell signaling. And this is very similar to communication. So the plasma membrane is best explained by using this structural model called a fluid mosaic model. And this model explains how the plasma membrane kind of moves according to what proteins are built within it. So this model, according to this model, the molecular arrangement of the plasma membrane resembles this continually moving sea of fluid lipids that contains this mosaic of a lot of different proteins. So you can think of the plasma membrane as kind of this flowing sea of water that goes up and down and flows very fluid-like. And if we add a lot more of these cholesterol proteins within it, it's going to become a lot more rigid and it's not going to flow as much. There are two kinds of membrane proteins that we're going to talk about. The first one are integral proteins. They're also called transmembrane proteins. And these are proteins that extend into or through the lipid bilayer, and they're pretty firmly embedded in it. Most of these transmembrane proteins, they will span the entire membrane and they're going to protrude on both sides. So kind of like this channel protein you see here. It's integral, so you have it opens onto both sides of the plasma membrane, as you can see here. So like membrane lipids, integral membrane proteins are very amphipathic, so they have to have a hydrophilic region on both sides of the membrane, and then also this hydrophobic region on the inside of the membrane as well. So these are also going to be amphipathic. The second protein that we're going to talk about are peripheral proteins. And like their name implies, peripheral proteins are not as firmly embedded in the membrane. They're really just attached to the polar heads of lipids or to integral proteins at the inner or outer surface of the membranes. So here you can see a peripheral protein. It's really just attached to the heads at the surface, and it doesn't go into this hydrophobic region on the interior of the membrane at all, so it is going to be a very hydrophilic protein. Membrane proteins largely reflect the functions that the actual cell can perform. First one we'll talk about are ion channels, and these are integral and they form a pore through which a very specific ion or a charged particle can flow through. So like I said, these ones are very specific. So for an example, they can only allow, one ion channel can only allow a sodium ion to flow through and nothing else. That's just an example. There are many different kinds of ion channels, but they can only allow specific ions or charged particles through. Carrier proteins, these are also integral and they transport very specific substances also across the membrane by undergoing a change in shape. That's the most important part about those proteins. The next one is a receptor. It is also an integral protein, and it recognizes a specific ligand. As you can see here, it has this very specific ligand. A ligand is just a molecule that interacts with a receptor, and the receptor will alter the cell's function in some way once it binds with that ligand. An example used in the textbook is that an antidiuretic hormone will bind to receptors in the kidneys, and it changes the water permeability of certain plasma membranes so that you're going to produce more water in your urine or less water in your urine according to how many receptors are targeted. An enzyme is going to be the next membrane protein we'll talk about, and this can be integral or peripheral, and it catalyzes reactions inside or outside the cell, depending on which site or which side of the cell the active site faces. Linker proteins, these can be integral or peripheral as well, and their function is to anchor filaments inside and outside of the cell, and these really work to provide structural stability and cell shape. So here you can see they're linking to those structural proteins on the outside and then also on the inside of the cell. And then we have cell identity markers. These are glycoproteins, and they distinguish your own cells from those cells from those cells of another. So a major cell identity marker is going to be the major histocompatibility complex. It's spelled out right here. And these are a very important type of cell identity marker that will identify your cells from foreign cells. Typically, there are 3 factors that bind cells together. So the first one that we're going to talk about are glycoproteins. These pretty much act like an adhesive and hold cells together. Wavy contours are going to be the next one, and they fit adjacent cells together in kind of a tongue and groove like fashion. And then we have special cell functions as well. Now with these factors in mind, there are 3 different types of cell junctions. We have tight junctions, gap junctions, and adhesion junctions. Tight junctions, these are very impermeable junctions, and they form this continuous seal around cells, and this prevents any molecules from passing through the intercellular space. So here you see the intercellular space is quite small, nothing really can pass between there. Gap junctions are communicating junctions, and they allow ions and very small molecules to pass through these pores or channels, and they're very important for communication in heart cells and in embryonic cells. Heart cells and embryo cells. Adhesion junctions, these are anchoring cell junctions, and they will bind adjacent cells together that act kind of like Velcro on the interior with these extracellular filaments. Membrane fluidity depends usually on 2 things. The first one is going to be the amount of double bonds in the fatty acid tails. And remember, this is the difference between saturated and unsaturated fatty acids. So saturated fatty acids are going to be all completely straight tails. They're going to be able to bind really easily between them. If you have an unsaturated tail, it's going to be bent, remember, and so you're not going to have as good binding or bonding between those tails of the phospholipid bilayer. So here in the unsaturated one is going to be a lot more fluid plasma membrane, whereas in the saturated tails, these ones are going to be very rigid because they're all going to be bound very tight together. The second part that fluidity is dependent upon is going to be the amount of cholesterol within that phospholipid bilayer. So membrane fluidity is a really big compromise for the cell. So a rigid membrane really lacks a lot of mobility, but a completely fluid membrane would lack the mechanical support and structural organization that is really required by the cell. So we kind of have a trade-off between rigidity and having that structural component of the cell and being fluid in order to be more mobile. The term permeable, this means that the structure permits passage of substances through it. While impermeable, this means that the structure doesn't allow any passage of substances through it. So the membrane itself, like I said, is selectively permeable. So that is that they let some things through, but not others. It's very permeable to non-polar molecules like oxygen molecules, O2, also carbon dioxide, and some steroids as well. Steroids are very permeable. And it's moderately permeable to some very small uncharged polar molecules like water or urea as well. So it's kind of slightly permeable to those. But oxygen, carbon dioxide, and steroids, very permeable to that. The membrane itself also is quite impermeable to ions and large uncharged polar molecules like glucose. Glucose is not able to pass through the membrane by itself. So these large macromolecules like glucose, they're only able to pass through the membrane by other means like vesicular transport. So these are going to be by vesicles. We'll talk about that a little bit later as well. So because the membrane is selectively permeable, this allows the cell to maintain different concentration of certain substances on either side of the plasma membrane. So the first one is this concentration gradient, and this is the difference between the concentration of specific chemicals on one side of the membrane as opposed to the other. In electrical gradient, this is the difference in the concentration of ions or charged particles between one side of the plasma membrane and the other. And together these gradients make up this electrochemical gradient. So this is the differences in both the concentration of molecules and the concentration of ions. Many ions and molecules are a lot more concentrated in either the cytosol or the extracellular fluid. For example, there's a lot higher concentration of sodium ions in the extracellular fluid or outside the cell, and there's a lot more potassium ions on the inside of the cell or in the cytosol. Typically, a substance will want to move down its concentration gradient or move from places of higher concentration to lower concentration. So say, for example, we have this membrane. This is going to be a pretty permeable membrane. And on one side of it, I have a lot of sodium ions, very highly concentrated with sodium ions on one side of the membrane, and on the other side of the membrane it's very low in concentration. We're only going to have one. So typically substances will always want to move from high concentration to a low concentration or move down its concentration gradient. That's just another way of saying, yeah, move down a concentration gradient, move from high concentration to low concentration. So these sodium ions will naturally want to move from left to right in order to create a homeostasis, to have the same amount of sodium ions on both sides. So then we would have, they would continue to move until we had the equal number of sodium on both sides. So these moved over here, and now we're going to have kind of this equilibrium. And when you have equilibrium, then you don't have any net movement of ions going either way. So these concentration gradients, when you do have a lot more of one ion or one molecule on one side of a membrane than another, you can use that because that's a potential energy, because naturally the ions will want to move down their electrochemical gradient, and the cell uses that to produce energy, which we will talk about a lot later. So transport of materials across the plasma membrane is really essential for the life of a cell. Substances generally move across cellular membranes by the transport processes that can be classified as either passive or active. So this is whether they need energy to move across the membrane. So passive, that's going to require no energy, and active processes require energy. And remember, the energy in the cell is going to always be ATP. This is the energy currency. So active processes require ATP input, whereas passive processes don't require ATP input. So we can kind of think of a membrane as water. And when we're thinking about active and passive transport processes, the water is going to be this membrane, and we need to cross the membrane. And we have this bridge, and this is going to be like a channel through the membrane. And if it's passive, the cars can just go straight over the bridge, no problem. If it's an active process, then you need to require to pay some money, like a toll, in order to go over the bridge. And that's going to be ATP in the case of a cell. You have to pay the currency in order to pass over the cell. Okay, here is a nice animation of transport processes across the plasma membrane. You can click on that on your own set of slides and go and watch that animation to help you better understand. And that will be the end of part 2 of our chapter 3.